invitation. I must say that this Max Planck Institute in Buenos Aires, I think, will benefit all science all over Argentina. And I found particularly promising the fact that the name Hiol is in the Max Planck, because Hiol is a family coming from Mendoza. This is indeed a winery, and Mendoza is one of the best regions in Argentina for wine. So I'm going to talk about dynamics of membrane remodeling during acrosomal exocytosis. I will set up my time here. Good. What we are particularly interested in my lab is membrane fusion as a mechanism, intracellular mechanism that is useful for many things, and it's very important for fertilization. You need membrane fusion during the exocytosis of the acrosomal granule, and you need membrane fusion for the interaction of the gametes to form a new organism. You need to release the content of the cortical granules to avoid polyspermia. So membrane fusion is central in fertilization. Now, membrane fusion is not a spontaneous process. You require quite elaborate machinery, molecular machinery, and this is a kind of comparison of this machinery occurring inside the cell. That these are snare proteins, and outside the cell by these viral proteins. But I must say that this comparison is more an exception than a rule. Fusion in the extracellular medium, where calcium is very high, is very divergent. Every process of membrane fusion among cells is different. Instead, inside the cell, the molecular mechanisms are quite well conserved. Just an example of the divergity of membrane fusion in cell-cell fusion, this protein, the pheromone-regulated membrane protein, are completely different than the membrane of the membrane protein that you require for egg sperm fusion in C. elegans, and completely different of the CD9 and isomos protein that you require for fusion in humans. And indeed, even in humans, different cell fusions, for example, the growing of a myotube or the formation of syncytial trophoblast, use a different mechanism. It's not isomo, it's not CD9. On the contrary, the fusion inside the cell is well conserved. And you have fusion, and you require membrane fusion for transport along the endocytic pathway and the secretory pathway. And I will refer mostly to regulated exocytosis that require calcium. It's a very well conserved mechanism from yeast to mammals, from neuron to hepatocytes. All cells use the same mechanism. And the mechanism, to simplify the mechanism, what you require is something to recognize the membrane that must fuse. And this is what is called tethering, and is arranged, is initiated by the recognition of RAB proteins. Not all fusions are good for the cell. You don't want a mitochondria to fuse with the nucleus. So RABs are like labels that tell what membranes can fuse. So, but this is not enough for fusion. Then you need this small membrane-associated proteins called snares, and the assembly of these snares complexes in trans force the opening of the connection between the compartment that need to be fused. And this is the components that are required in C. elegans, and you see there is RAB, RAB effectors, the snares, and snare regulators. In mammals, it's the same story. Now, what is our model? Our model is acrosomal exocytosis. So it's a regulated secretion. This is a human sperm. This is the acrosome. It's a flat granule, completely different than a secretory granule that you can think of synaptic vesicle, for example. And fusion occurs not just by opening of a single pore. Hundreds of pores are opened, and that causes the destabilization of all these membranes and the formation of these vesicles that are called hybrid vesicles. The mechanism is not well understood. If you think in two dimensions, and if you have two membranes and you open a couple of pores, you can think of the formation of hybrid vesicles. In 3D, it's more difficult. If you have two membranes and you connect with two pores, that will not produce a hybrid vesicle. So it's more complex than that. This is the sperm after the acrosomal exocytosis. Now, a new plasma membrane or a new membrane is surrounding the sperm. This is the remaining of inner acrosomal membrane. This is the remaining of the granule. And this is very important because that changed the composition of this membrane. And this part over here is the one that will interact and fuse with the egg. So as a summary, acrosomal exocytosis means it's just a single, very large granule that you can see in a regular microscope. There is a lot of fusion pores. There is not intracellular transport. So the only fusion event that will occur in the sperm is this exocytosis. And there is a problem. There is no transcription or translation. So we cannot overexpress protein. We cannot knock down proteins. What we have done in the last 10 years, I think, was to develop a way to introduce proteins inside the sperm, and that was done by streptolysine that then we can add to this system recombinant proteins, antibodies, toxins, or whatever we want to get into the sperm and to interfere with or to interact with the fusion machinery that is there. Not surprisingly, what we found is the same kind of protein that we were expecting. Now we know exactly what proteins are required: RABs, RAB effectors, members of the three family of snares. What is interesting that these snares are brain snares. They are neuron snares, sensitive to neurotoxin. That is very important for us. And all the snare regulators, except MANC18, and we have the evidence for this protein, but it's still not published. Not only that, but sperm is a great system to study exocytosis. So we have a quite complex pathway for the interaction of these proteins. And what we can say, to summarize, is that we require a branch of assembly of proteins, snares and snare regulators. But we also need a branch we call the lipid branch, because we need to produce several lipid intermediate, and the final end is to produce IP3 that will open calcium channels in the acrosomal membrane. A simplification of this process is here, just showing just a few proteins. What we think that happened is when calcium gets in the sperm, it's able to activate all the machinery that in the sperm is waiting for this calcium. Synaptotacmin is phosphorylated. NSF is inactivated by phosphorylation too. Snare are pre-assembled on cis complexes, inactives. Calcium will trigger the activation of all the system. Snare will now interact in trans, and this requires complexing to stabilize this. And the system will stop there. Will stop there until calcium is released from inside the acrosome. And that will activate synaptotacmin and then will open the fusion pores. But then, all this biochemical characterization led us to some prediction, which is that we can test by electron microscopy. What is the prediction? If we inhibit the release of calcium from inside the acrosome, the system will stop here. And here, the snares are pre-assembled in transcomplexes, and the distance between the membranes cannot be more than 8 nanometers. So we did the experiment in the presence of this blocker of IP3 calcium channels, and what we observe is that now the acrosome is swollen. There's vesicles inside the acrosome. There is a waving of the outer acrosomal membrane. And if we look for apposition, we found tight appositions of the membranes at the edge of these invaginations of the outer acrosomal membranes. This is more dramatic, showing the two membranes in tight apposition. And there's one more here. And our model now say that if you open a fusion pore here, this invagination of the outer acrosomal membrane will form a hybrid vesicles. This is well shown here. This is a hybrid vesicles. Fusion pore must open here, and that will release all these hybrid vesicles. This is a theory, and so the prediction is that anything that produces exocytosis in the sperm should provoke swelling, and this is the case here. Swelling can be regulated. Aquaporins are required, and some ion channels are required. Whatever, if we inhibit swelling, we inhibit exocytosis. And another prediction is if this apposition requires snares, if we do the experiment in the presence of toxins, the membranes will never reach these tight apposition conditions. That's the experiment. We measure the distance between the membranes, and this is our control. This is an histogram of the frequency of membrane distances, and the more frequent one is less than 8 nanometers. If we add complexing, that would favor this, the frequency of this tight apposition increase. Instead, in the presence of the toxin, it's significantly decreased. And this is the work done by Natalia Zanetti. She's doing a postdoc. She had twins now. That's very dangerous to manipulate the sperms. What I will just summarize is saying that besides these two branches that we had characterized before, we need to have another set of proteins that will be involved in the acrosomal swelling and working in that problem. The deformation of membranes, the transport of water in and out of the acrosome. The story is still quite complex. The lipid branch require a lot of different proteins because it's required to generate PIP2 for a continuous production of IP3. And diacylglycerol can crosstalk to the protein branch. And this is a work that is being studied by Silvia Belmonte and her group, Leo Peletan, Silvia, Laila Swayman, and Cecilia Lopez. And in the protein branch, it's also quite complex. This is recent evidence that MANC13 and RIM are required and when are required during exocytosis. And this is a work that Claudia Tommy is performing with her group, Matias Bustos, which is in the audience, and is working in RAB27, another RAB that is required for this exocytosis, Oscar Bello and Facundo Rodriguez. Now, in the last few minutes, I will talk about new strategies. The problem with our model is that we kill the sperm. It's very nice, but a permeabilized cell is a dead cell. So in this permeabilized cell, and we are using here, instead of streptolysin, another toxin that is cheaper, and they work very nicely in our system. You see that calcium that cannot go through membranes here triggers secretion. The same that RAB3A when it's activated with GTP gamma S. In intact sperms, instead, calcium and RAB3A has no effect. A few years ago, we tried to make a RAB3A that can go through membranes, and we hook a small peptide in N-terminus of RAB3A. This polybasic peptide now allows RAB3A to go through membranes. And this is a functional assay. You can see that this variant of RAB3A can trigger secretion in intact live sperm. No? And this is the control that the protein must be active to trigger secretion. So we are now interested in the process of capacitation. What is capacitation? The sperm, when it's ejaculated, is not able to fertilize an egg. It's required to go through the female tract, and something happened during this passage through this tract that prepared the sperm for exocytosis and for fertilizing the egg. We always work with capacitated sperm, and we know that in capacitated sperm, snares are pre-assembled in cis complexes, inactive. Cyanotactmin is phosphorylated, NSF is phosphorylated. Now, we wonder if this very particular condition of the fusion machinery is occurring during capacitation. For that, we cannot use permeabilized sperm. These are one of the experiments you can perform for testing, for example, that the snares are protected in capacitated sperm. You treat with toxin and nothing happens. When you inactivate the toxin and you stimulate secretion, it's normal secretion. We can test this by immunofluorescence, since the toxin what does is to cleave VAM2. We can see by immunofluorescence that it's not a fact. Now, if we use membrane permeant tetanus toxin, we can see that in non-capacitated sperm, the snares are sensitive to the toxin. So something occurred during this transport, and this fusion machinery is maturating this transport. For that, we need this membrane tetanus toxins, and we can ask a question when the snares are protected, and you require about 2 hours of capacitation to get to lose the sensitivity to the toxins. Living with or studying live sperm is our goal then. One other thing that we want is to see if we can detect acrosomal exocytosis in live sperm. And the strategy was to add some compound that has high affinity for the content of the granule. In this case is a soybean trypsin inhibitor. When the fusion pore opens, this probe can go in, but then when the acrosome dissolves, the probe is lost. If you look at this sperm, it's the first one that is going to take the soybean trypsin inhibitor, see, and it's gone in a few minutes. For our surprise, when tried another probe that should work the same, that is a lectin, a fluorescent lectin, we observed that at the end of the experiment, all the reacted sperm were green. And if we quantified this in different sperms, this with soybean trypsin inhibitor goes up and then down. With the lectin, it stays forever. And we wonder why. And the reason is that the lectin is like a glue. It will keep all the vesicles, the hybrid vesicle, attached to the head of the sperm. It will never be released. So that's why the sperms stay green. And This is with a little more details. See the nuclear envelope, the inner acrosomal membrane, and the hybrid, the membrane, the hybrid vesicles. The lectin do not affect exocytosis, but do affect the number of sperm that retain these vesicles. Well, this is an observation that is nice because will allow us to study with more details the process of vesiculation. It's also interesting because allow us to sort live reacted sperms. And this is done by FACS, and you see a population of green sperm coming up with time. And this is a double labeling to detect dead cells. And you see that the population that we got are live green sperm that have reacted. So we can sort them and look in the confocal microscope. And now we have this population, and we can ask questions like why some population of sperm, only about 30% of the sperm can undergo acrosomal exocytosis, and the rest remain with no alterations. This is the work done by Martin Sopino, working with the FACS and confocal, and another Martin Pavarotti, working with the ferment proteins. And with this, I would like to acknowledge the agency supporting this work, CONICET, Agencia, Universidad Nacional de Cuyo, and part of this was also done during sabbatical I did thanks to a Humboldt Award in Heidelberg, and that's allowed me to bike in Heidelberg. It was a great experience. It's a beautiful place. You cannot believe this. Okay, but I'm back in Mendoza, and it's also beautiful. Thanks for your attention.
You can raise intracellular calcium in sperm by different means. For example,